Hey Paula, this is Nate Powell.
Very official.
Next to me.
Oh, I thought it was my phone.
Paula, you hear me?
Yeah.
Um, are y'all,
With us.
are
Oh,
y'all at, like Green Fisheries right beside of IMS right now?
Yeah, it's good.
Say, Nathan.
I'm not a.
Yeah, are you just next door at Marine Fisheries?
Down in the central business office, nation.
Oh, is that the one down further down Rendell?
Yeah, it's across 24.
Towards Newport.
Yeah,
I thought you were out of town.
Well, I was planning on being out of town, but then I decided to come back and be in person, but then I thought it was next door, so I messed up. I still would rather be there in person though. If it's all right with y'all, I'll just drive down and I'll be a few minutes late.
That'd be great. Thank you, Nathan.
Okay, I'll be down there in a minute.
I like it. It's very southern.
No.
We are now. Weather service.
Oh.
Problem. There's still some like
So, can you hear us?
some other apps.
That's one of my apps. Sorry.
It's provided by the way.
Predict wind is much better. But I usually hear it,
Part of it. Yeah.
And the boat you asked. I think that's
I
it.
know.
No, this boat you guys is the one.
I'm in a good spot because I can't have here.
They might use it, which is
I think they do. And then obviously the NOAA app.
we don't have an app.
Yeah,
Good. So this app,
they are.
You go to it.
I could hear Nathan, like.
Yeah, I
here,
could
but-
too, but it's a lot of the.
again, I use a whole bunch of apps. This isn't you, Don?
So are you here?
Go to your app store.
That's not you.
How you doing? It's good. Maybe
Even though it says NOAA weather.
probably just use it. So it's just,
Okay.
but as far as like. we don't-
And just to also make sure.
Yeah. Check your audio.
Checking my audio. Can you hear me? This is Lisa.
Yes, we sure can. Thank you.
Okay, thanks.
Hear us?
Oh,
Let me get
it's
your.
because it's available. But I'm almost, positive this also says. No.
Well, we can make note and just post it on like our attendance sheet, so we won't call you Jack.
Okay.
All right, it's April 19th at the Habitat and Water Quality Standing Advisory Committee. We're gonna do a call to order. And we have some new people in our midst, and I'd like to let them introduce themselves. So, can we start with you?
Sure. All right. So I'm David Glenn. Was on the committee several years ago, took a little break, and now I'm happy to be back. I work for the National Weather Service in Newport, just up the street. But yeah, look forward to participating and however way I can.
Bart?
Mhm. I was here the last time at the,
Okay. I
at the virtual.
Okay, it's, all right.
Yeah.
Sorry.
I go by Bart.
I'm sorry.
Tax my name they made me put on the application.
Gotcha. And I'm sorry to not recognize just the virtual thing threw me off.
Okay.
I apologize.
I'm Captain Mark Sonder from H2O Captain Echo Tour Private Boat Excursions. Glad to be here.
Nice to meet you. Can we? Yes.
Hi, Scott Leahy, work over at Carter Community College for the maritime programs.
Very cool.
Can we vote on the? Oh, yes, online folks want to join in? Doug, can you hear us? Can you say hi?
Hi, everybody. Doug here. Co-chair with Anna.
Got Lisa Ryder.
Hey, everybody, Lisa Ryder. I may have weird connections. I'm actually volunteering over at the Cape, so I got the lighthouse behind me. And really happy to be here online with you. Thanks.
All right, okay, do we have a vote on the approval of the minutes for this evening? Everyone here say aye?
Thank you. That's right.
Does anyone want to make a motion to approve the minutes?
Motion approved.
Thank you. You can tell that I'm new. I apologize.
I'll second
Thank you. Thank you. All right, we do not have Laura Kilbansky. And we vote for the, I'm sorry. I'm embarrassed. I'm incredibly embarrassed. All right, well, now we're going to vote for the approval of the minutes. All here say aye.
Aye.
Aye. Laura Kolbansky is not here. We're gonna have Paula fill us in with the Marine Fisheries Commission update and the July joint meeting planning. And pardon my newbiness.
Thank you, Anna. Okay, switching over here so folks can hopefully see me. As Anna said, Laura Klebansky is not here. I think I've met / interacted with most of you obviously at our January meeting, but I am Paula Farnell. I'm the program assistant for the Marine Fisheries Commission office. So I work side by side with Laura. So I'm gonna give us our update for tonight. So the Marine Fisheries Commission's February business meeting was held in New Bern. And I'm going to give you a brief overview of highlights from the meeting, but we just want to remind you guys that the meetings are recorded, just like these advisory committee meetings. They are available to watch on YouTube through our website. Our public information office staff do a great job of what I call chaptering those meetings too. So if there's specific subject matter from within the meeting that you want to watch and review, you can click right through into those specific agenda items or chapter sections. So it's easily available for you there to review more things in detail if you would like. We do just want to let you know that it will impact your YouTube algorithm. So if you go back and watch too many meetings, you might start seeing some interesting DEQ topics come up on your YouTube suggestions there. So just FYI. So we're going to start off with false albacore. In February, the commission reviewed a false albacore information paper, which the division had prepared at the request of the commission. And this was an update to a 2017 paper that was a general review of information about the false albacore fishery in North Carolina. Following quite a bit of discussion on the information in that paper, the commission ultimately did pass a motion asking staff to develop rulemaking language with management options for false albacore, starting with the status quo and allowing for growth in the fishery at various percentage points. So staff have spent time between the last meeting and our upcoming May meeting reviewing available data to define some of those terms. For example, what is the status quo, as well as looking at some of those potential percentage points. And so there will be new information, that initial analysis presented at the May Marine Fisheries Commission meeting. And there will be time to discuss that status quo information and what staff anticipate the various percentage points to look like. And then the final issue paper with the rule language options is anticipated for either the August or November commission meeting. We're going to move along to spotted sea trout. So in February, the staff leads presented an overview of the spotted sea trout fishery, and they received input from commissioners on items for consideration in the FNP development. And just as a reminder, we just completed the scoping period for spotted sea trout. So we're at the very beginning of FNP development. There is no draft plan or anything of that nature. And as I think you're all aware, there's actually going to be some time later in this agenda to talk a little bit more about that scoping process. But basically staff are now going to be taking that feedback from the scoping process and from the commission and use that to begin developing the actual draft FNP. And we will also have the opportunity during that discussion with the spotted sea trout leads to be able to provide your feedback as this committee as part of that process as well. So we do want to acknowledge the feedback that was provided by Commissioner Cross. You may or may not be aware of that, depending how closely you follow some of this process, but it was a lot more comprehensive information than we sometimes receive at this stage in the process. But it did allow us to be able to receive some very clear public comment on those types of thoughts in terms of management. And it really did help us get the conversation started. So it turned out to be a good thing in the long run that we received those comments, at this point in the process and be able to utilize those as we move forward with the plan development. And we just want to point out that, public input is a huge part of fisheries management in North Carolina. And so any conversations that we have with the public are a good chance to be able to talk about that process. And the sooner that we have those conversations in the process, the better overall. So moving along to striped mullet. Back in November of last year, the commission selected their preferred management option for the striped mullet supplement A. And that was for statewide November 7th to December 31st season closure, which is estimated to result in a 22.1% reduction. In February, the commission heard the outcome of the public comment period, which was based on that chosen option for supplement A. And based on those comments, they requested the division consider developing regionally specific seasons. And so staff are currently working on that, and they will present updated information at the May commission meeting. And just as a reminder, a supplement is meant to address overfishing immediately while more comprehensive management is developed in an actual amendment. So in May, the commission will be continuing their discussion of the management options in supplement A, and they may either select from the original options, which were presented and discussed in November, or from new potential options which staff present to them during the May meeting. And finally, I'm going to wrap up with an item from the Coastal Habitat Protection Plan. Relating to the CHIP, the commission unanimously approved a motion supporting the Coastal Habitat Initiative resolution. And that came out of the Stakeholder Engagement for Collaborative Coastal Habitats Initiative, otherwise known as SECI. And this particular resolution was focused on encouraging the state to increase funding for voluntary cost share programs, which help improve water quality. So as a reminder, DMF and APNEP worked collaboratively with the core team of NGOs to form a public-private partnership, which is that Stakeholder Engagement for Collaborative Coastal Habitats Initiative. And it was a recommendation directly from the 2021 CHIP amendment. And so the resolution that was voted on by the commission was also supported by Coastal Resources Commission and the Environmental Management Commission at each of their recent meetings as well. And the May commission meeting is scheduled for May 24th to the 26th at the Beaufort Hotel in Beaufort. And I've so far talked about striped mullet, spotted sea trout, and false albacore, which are all on the May agenda. But if you'd like to see a more overview of what is expected to be on the agenda, you can always review the MFC work plan, and that is always made available within the meeting materials for each MFC meeting. And so those meeting materials are typically released 2 weeks before the actual meeting date. They'll be available on the website. That MFC work plan is also often sent out to you guys, so we can also send an updated version as it does get updated between the various commission meetings, and it's kind of a rolling working document. But that's a great way to keep a longer term view and track of where things are at in this FNP process, because it usually does about a 3 to 5 year timeline, depending on what all things are in process at the time. And then if people have questions specific to the commission or the commission meetings, I can take a break here. Otherwise, I can roll on into the July AC workshop conversation and we can kind of handle all the questions, whichever works better for you guys.
One thing I may add is on the, that resolution, it was also approved by the Soil and Water Conservation Districts Commission,
Okay,
which wasn't, we didn't know that at the time. And several other entities, organizations have signed on to support it as well.
Awesome.
So if anybody here is associated with an organization that you think would support efforts to improve water quality through, improvements for farms, just let us know and we can reach out to them and see if they want to sign the resolution. They said the more diverse the stakeholders, the better for increased chances of getting funding.
Thank you, Ann. Appreciate that. You are the one in the know on that for sure. Had to work here tonight, so thank you. Any other questions or comments for the MFC related updates? Okay, I'm going to move along into just a super brief discussion about the July AC workshop. So you guys are aware we had sent out some doodle polls and some surveys and questionnaires to you guys to talk about potential dates for this workshop. And again, this workshop is, something meant to be able to bring you guys together in person, which has been a request from several of the committee members across the committees. So we have decided on Monday, July 10th, based on the questionnaire results, over 50% of those that responded chose that as one of their top dates. We recognize that will not accommodate everyone, unfortunately. We're trying to satisfy 60+ people who are all across the state and have all varying types of schedules based on all of your jobs, that you work full time and you volunteer to be on these committees. But so that was the best date out of the choices. And it will actually take place at the North Carolina Aquarium here at Pineville Shores. We also recognize that's going to require travel for a lot of folks, but we were able to work with the aquarium and have that space for a reduced rate, which will hopefully allow us to provide for hotel stays and for travel reimbursements for folks that do have to travel to the meeting. Just a reminder, as I think some of it got lost in some translation, that we are looking at like a full day type of thing where it will be from 10 a.m. to 3 p.m. as this workshop is designed to include multiple presentations and multiple opportunities for facilitated discussion for the committees to really be able to work together. But we will be sending out further details in the coming weeks. So we just wanted to give you guys the update in terms of the date. And, again, just the concept being for collaboration. We have various committees that cover various topics and regions, and we have various folks across these committees with various levels and wealth and depth of knowledge on various topics. And this will be a great opportunity to bring folks together. We have several new folks across all the committees, in this particular go-round opportunity there to bring up some information that you have questions about. Kind of going to be general topics, things such as like stock assessments and the FNP process and that advisory committee's roles in that FNP process and how that relates to the commission and sort of things of that nature. We're still fleshing out all the details, provide those presentations and around those presentations, ability for you guys to also discuss, these things amongst yourself and provide your feedback in some of those processes and any questions and comments that you guys have and be able to interact some more with some of the staff and things like that. So really just a chance to have discussion and collaboration amongst the committees.
Awesome. Thank you so much, Paula. Next, we're moving on to spotted sea trout scoping period discussion. There is no presentation, but I'd like to open up the table to a discussion about spotted sea trout. If anybody has any questions or concerns that they'd like to bring to the table. Lucas is putting an update.
Well, yeah, I can update.
Get it.
Ooh, I'll pass on that one. Tempting though, tempting. So good evening, y'all. We appreciate the opportunity to come here and talk about some of the comments we heard during the scoping period for spotted sea trout. And also to hear your input tonight. I'm Lucas Bentinger. I'm a biologist here in the Morehead City office, and I'm one of the co-leads for the spotted sea trout fishery management plan. Jason Rock is also obviously up here tonight. And he is a biologist supervisor out of this office, the other co-lead for the FSC. So the scoping period for Amendment 1 of the Spotted Sea Trout Fishery Management Plan was from March 13th through the 24th. We actually had over 700 people participate in that process, scoping process, by either coming to a meeting or submitting comment through our online questionnaire. So it really was awesome to see that many folks get involved in the process. Sometimes a little chaotic, but awesome nonetheless. So for most of the topics that were brought up, comments varied from strongly for these topics, strongly against these, and kind of everything in between. So it really did run the gamut there. So I thought I would start tonight just by giving a brief overview of the comments as they relate to each potential management strategy. And then we'd obviously be happy to answer any questions. And also we'd love to hear your input on potential management strategies, both on the ones that we've already kind of identified and any other ideas that you might be a little more outside the box. Well, I will say we've heard a lot of outside the box. So I'll start with sustainable harvest. Comments around sustainable harvest centered on not implementing a quota in the spotted sea trout fishery. That's one of the few that there weren't really both sides of that one. Folks are pretty strongly against implementing a quota. Seasonal closures, a bag limit reduction or a trip limit reduction, and increasing the minimum size of spotted sea trout. For recreational management, I'll kind of split those into spotted sea trout specific and then just more overall general recreational management. As far as spotted sea trout specific comments, those centered around making spotted sea trout a game fish, outreach, especially around catch and release best practices, eliminating the recreational commercial gear license as a means of harvesting spotted sea trout. So that would basically just mean that folks can't fish recreationally with commercial gear. Implementing boat limits, eliminating the captain and crew limit, and then just a general limited entry into the spotted sea trout fishery. So the more general things we heard, and these are more general just because they would impact more than just spotted sea trout. So they would probably have to be a broader conversation across multiple fisheries. So for that one, we heard comments about reducing the impact of tournaments, especially catch and release tournaments, and then implementing a variety of gear requirements. So things like circle hooks if you're fishing with live bait, getting rid of treble hooks entirely, getting rid of treble hooks on artificial baits, things along those lines. For commercial management, I also separated that out into spotted sea trout specific and the more general stuff. For spotted sea trout specifically, we heard comments about making the spotted sea trout fishery or commercial fishery entirely a hook and line fishery, and then also comments about limiting entry into the spotted sea trout fishery. For the more general comments, those comments centered around reducing gillnet efforts, reducing all commercial effort, closing the personal consumption loophole, offering commercial subsidies in order to phase out the use of gill nets, setting some area limits, so like limits of where folks can fish commercially, increasing gill net mesh size, and requiring gill net attendance regardless of time or area that folks are fishing. As far as protecting spawning stock biomass is concerned, we heard comments centering around implementing slot limits, reducing the bag limit, increasing the minimum size, and then a variety of comments around our cold stun closures, especially with those, with the slot limits and the bag limits and the increasing minimum size. Comments specifically for protecting spawning stock biomass were more around some temporary measures. So if we close the fishery due to a cold stun, when it opens up that year, having a temporary slot limit or temporarily reducing the bag limit, things along those lines. Although again, if we go back to sustainable harvest, we also heard a lot of just general call for those things. As far as area management is concerned, this is actually one that we identified throughout the scoping process. It wasn't necessarily on our radar at first, but we heard a lot of calls or we heard a lot of comments, I should say, centered around closing specific areas to gill nets, closing specific areas to all spotted sea trout fishing, so recreational and commercial, and implementing a more regional or localized management strategy. So we kind of added that one after the scoping period, just because we heard a lot of comments about that. To speak specifically to Commissioner Cross's proposal, as Paula mentioned, he did get us that proposal in enough time that we could take it out during the scoping process and see, get the public's input on it. And I will say this is another one of those few areas where comments were pretty overwhelmingly in one direction. And really comments were overwhelmingly against that proposal and felt that it was unnecessary in the spotted sea trout fishery at this time. Specifically, those comments were, again, going back to no quota. We heard a lot of comments about not implementing a quota in the spotted sea trout fishery. And we heard a lot of comments about not ending catch and release fishing. So there were some ideas in that proposal that would call for removing gear from the water. And we heard a lot of comments that people were not interested in that. And then there were a couple more general ideas that really didn't fit into any of those categories. And those centered around more ecosystem or multi-species based management. So considering how, any spotted sea trout management that we put into place might affect fishing for other species. Also around stocking fish, increasing enforcement efforts, looking at management in other states and implementing what might be working there here, and then around a recreational reporting app. And for that, we heard folks wanting that to be a voluntary process. Also heard folks wanting that to be a mandatory process. So that, is I think I'm gonna steal Jason's joke from last night. Might not have felt overly brief, but that is definitely a brief rundown of the comments that we heard during this process. So I'd be happy to answer any questions y'all might have, or we'd definitely be happy to hear your input as well.
I got one. One of the things so I teach over at the college for our community college here, captain's license courses. So I get a lot of potential charter captains coming through. And one of the points of interest that came up was the discussion about the discard mortality rate. And I'm like, and overwhelmingly, they're all saying that, hey, I never throw away a dead fish, of course, is what they're claiming. How was that data captured to reach that high mortality statistic?
So to start with, I will say that we do call it delayed mortality for a reason. That is a lot of times that discard mortality can be more obvious where you do throw away a fish.
Right.
It goes belly up immediately. But it can also be a lot less obvious where the stress of that fishing interaction causes mortality, a day, 2 days, potentially 3 days even later. But that rate, we came up with that through a study that was conducted right here in North Carolina. So they went out and fought spotted sea trout through, and there were other species in the study too, the spotted sea trout specifically, through a variety of methods common in the recreational fishery, so bait, artificials. Caught the fish, identified if there were any hooking injuries, where they were hooked, all of those things, held them in a pen. I think it was for 72 hours, I want to say. And then just in I think 12-hour increments or 24-hour increments recorded mortality. So which fish died, which fish didn't die. And we're able to say, coming out of that, we were able to estimate an average mortality of 10%.
Okay.
Now recognize that, an individual's, my personal discard mortality rate might be higher or lower than that. And a lot of it depends on time of year, water temperature, how handling, how you handle it, and things of that nature. But in the end, that all averaged out to that 10%.
Cool. Thank you. Yeah, because that was one that's like no one could explain at the time when it came out, and that was just a big question mark on the data and the science behind it. So
yeah.
Thank you for that.
Yeah, and a lot of that was just time. we were trying to cram a
Oh, yeah.
lot of information into a
Yep.
short amount of time. And yeah,
But I will share
we
that.
got a lot of questions about that too. And I will say also, like, if folks have more questions, send them my way. I'm happy to explain it. Now, we spent a lot of time on the phone during scoping for folks that did call and had questions exactly along that.
Excellent. Does anybody else have any questions?
See, Doug had one.
Doug?
I think you're muted, Doug.
Must have double tapped it. Sorry about that. So one of the questions is whether you received any comments about habitat or water quality issues related to spotted sea trout, and particularly to the various life history stages, which I think we talked about before, and I would bring back up here in a minute about the degree to which different life history stages of spotted sea trout are vulnerable to either habitat or water quality issues that might be relevant in terms of estimating future abundance or management success. I just wondered what you heard from the public during the comment session.
I know that we heard much on that front, really.
Anything specific to habitat or water quality coming up during the scoping meetings?
Yeah, and so the reason I ask is since these are estuarine dependent, this is an estuarine dependent species, and they're estuary and primary and secondary nursery area users, the ability of those habitats to sustain productivity is a question, right? So the question back to you, and that we'll be asking, this advisory committee will be asking during this process, is there a degree to which we know whether or not that those habitat needs are being met in terms of sustaining the life history and ecology of this species?
I think I would, purely based on the spawning stock biomass that we see, I would lean towards yes, those needs do appear to be being met. The issue that we're seeing with spotted sea trout right now is an issue of removals. People are taking a lot of them out of the population, but it's not an issue of spawning stock biomass. There are a whole lot of trout out there. Spawning stock biomass is actually is well above the threshold level. It's actually well above the target level. So it's pretty high. And to me, that would imply that those those needs, those nursery areas are being met. I don't know that we have anything more specific than that necessarily to answer that question, but it certainly seems to be.
And so a follow-up, if I could, and so that would be based on current conditions. And if you then look at what happens as sea level rise continues to unwind and water is warm and populations move, then what, in particular, given the lead time in terms of restoring, both protecting and restoring those kinds of functions, whether or not there's a bottleneck to come in terms of that productivity. And so one of the questions I would ask would have to do with the spawn recruit relationships and the year class patterns, and whether there are any signals in that explains the cloud of points in terms of things that might then also in the future suggest that more would need to be done, since we need to be 10 years or 20 years or more ahead of that particular curve. And I guess I, at least, or I hope we, would like to see those kinds of issues laid out and examined as this F&P develops.
Yeah, and I think we'll get into some of that when we develop the plan itself, especially in the background section where we talk about habitat. I know you mentioned like sea level rise, with sea trout being, highly dependent on seagrasses, there's some potential there for there to be some negative impacts because of that. Same with marsh habitats. They're big users of marsh edges, and we know there's already seen some impacts to marsh edge habitat in particular. So right now, things do seem to be okay. Recruitment we've seen the last few years is some of the highest we've seen through the time series. So there doesn't seem to be a recruitment issue or a habitat issue at the moment, but yeah, like you said, in the future, that certainly could pop up, just depending on how quickly things progress.
I'm pretty sure, Doug, that the NOAA's climate vulnerability assessment that we're looking on that final report, I think spotted sea trout was one of the species they looked at. And so they rated how vulnerable the species would be with these changing conditions.
They were, and I should know where they rated out on that. I was involved in that process, but it's been a while since I looked at it. So.
Great. Well, I think it'd be nice, in fact, to look at like some of the other estuarine species assessments where they look at different life history stages and their whether or not, and then the relative scoring for habitat and or water quality vulnerability for different life history stages, just so that that's on the record. And I think we'd be interested. I'd be interested. I hope it's we in making sure that kind of information is laid out to the public now. And then as questions develop, and particularly things that we think together might be important and that we don't have the evidence that we need to be able to make a clear determination, that those things could also be captured in terms of potential priorities for research and for the development of new ecological science, particularly climate response science for this species and others like it. Well, I'll get off my soapbox, but I'm real interested in doing whatever we can do to help on making sure that that kind of information is well represented.
Thank you so much, Doug. We are moving on to shellfish sanitation presentation from Shannon Jenkins and Amy Hammond. Yeah, thank you guys so much. Thank you both.
Shannon.
I do. Yes.
All right, cool. All right, well, thank you guys for the opportunity to come out and meet you today. Apologies in advance for my voice. It's hanging on by a thread here, but we'll get through it. I'm Haynes. I'm the Growing Areas Program Manager with the Shellfish Sanitation and Recreational Water Quality section in the Division of Marine Fisheries. And I'm excited to talk with you guys a little bit about our program and who we are and what we do, and in particular focus on some of the water quality aspects of our program and how that relates to shellfish classifications. So one thing we like to always kind of emphasize right up front is that our mission is a little bit different from most of Division of Marine Fisheries in that we are primarily a public health agency. We are focused on kind of two main goals. First, to assure that all shellfish that are going to be harvested or processed in North Carolina are going to be safe for human consumption, and also to make sure that coastal recreational waters are going to be safe for human activity. Obviously, the recreational water quality portion of our program has a large water quality component given the name, but I'm not going to talk about that too much tonight. I'm going to focus primarily on the shellfish side of our program and the water quality aspects of that part of the program. But we are certainly happy to come back if you guys have an interest in learning more about the rec water quality program. So a little bit of background. A lot of you are probably already familiar with this, but a little bit of background on kind of why we exist. So shellfish, as you all know, are filter feeders. And as they feed, they can't particularly be selective in what they take in. So they not only take in the good stuff they're trying to eat, but they can also take in toxins or pathogens or other contaminants that might be in the water. And as they feed, they can also concentrate those contaminants within their digestive systems to a pretty significant level. And because shellfish, and particularly oysters, are a pretty unique product in that they're one of the only animal food products that we eat alive, whole, and raw. It provides some unique challenges when it comes to food safety and making sure that people are not going to get sick from consuming this food product. We need to basically make sure that it's coming from really clean areas, since there's not going to be a cooking process to kill off any bacteria or viruses that might be there, and that there's not any kind of, you're not cutting out fillets or anything like that. You're eating the whole animal. So anything that's in there, you would consume if you consume the shellfish. Shellfish sanitation programs like ours started in the 1920s all around the country following outbreaks of typhoid fever that were linked to tainted shellfish being harvested primarily near untreated sewage discharges like this one on the screen. People quickly learned that we needed to be a lot more careful with how we were, where we were harvesting our shellfish from. And currently, North Carolina is part of the National Shellfish Sanitation Program. It's a cooperative program between state agencies like ours, as well as the federal government and representatives from the shellfish industry who work together to set minimum public health and safety standards that all states need to meet in order to participate in the interstate commerce of shellfish. So the shellfish sanitation portion of our program has two main components. The first is the growing areas portion of our program, which is focused on the shellfish while they're still in the water. And then the inspections part of our program that is focused on basically everything from the time the shellfish are harvested until they're sold to the consumer. Again, I'm going to focus on the growing areas portion of our program today, because that's where all of our water quality activities take place. So we've got the whole coast divided up into a series of smaller management units that we call growing areas. These are basically kind of administrative designations that help us to divvy up our workload when it comes to monitoring and setting the classifications in these areas. The growing areas are also used for tracking of harvested product from when it's removed from the water and moves through commerce to final sale. In case of any illness outbreaks or anything like that, we can trace it back to where it was harvested from to make sure we can find where the problem arose in that process. We've got 49 growing areas that we actively manage currently, and then a number of other areas like you can see up in Albemarle that are completely prohibited, and we don't do any management activities there currently. There are two primary components to the growing areas program that we use to help set our harvest classifications. The first is the shoreline survey program. This is the part of our program that occurs primarily on land, where we'll actually go out into the field and visit all the properties in and around the watershed of each growing area in order to try and identify and assess any potential sources of pollution that could be impacting water quality in the growing areas. So we'll visit things like on-site wastewater systems to make sure they're working properly. Visit wastewater treatment plants to look at their structure and function, make sure they're not putting out contaminated water, and make sure they're not going to collapse or leak or cause spills. We also work with local health departments and Division of Water Resources on those inspections to make sure that we have a good comprehensive overview of how everything is working. For wastewater treatment plants and other point source discharges of pollution, we can also do hydrographic studies. These are studies where we actually inject the fluorescent dye into the waste stream as it heads out towards surface waters, and we can track that dye around in the shellfish growing area to kind of see how potential contaminants from like a wastewater treatment failure or another spill might dilute and disperse around the growing area, help us to determine in the event of an issue what type of closure we might need to make. We also look at animal sources, in particular proximity to the water and waste handling practices. We look at marinas, not only for human or animal waste from onboard toilets or from flocks of birds that hang out around the marinas, but also for chemicals and heavy metals that might be generated during boat cleaning or maintenance or repair. We look at development within the watershed, which obviously can serve to concentrate potential sources of pollution in a smaller area than where they were before, and can also increase the amount of impervious surface in the watershed, which ties into stormwater runoff, which we also do a pretty detailed assessment of the stormwater drainage network within each growing area to kind of see the size of the network, how much water is moving through it, the types of things that might be carried through that stormwater system, and how they might be impacting growing area water quality. And we take all of that pollution source survey information and import it into a GIS. You can see kind of just an example screenshot of one of our GIS setups here. Each of those individual colored dots on that map represents one particular pollution source that we visited and assessed. We have to do these detailed pollution source surveys once every 3 years for each growing area on our coast. So this GIS has a pretty good historical network of information from each time we visited each of these different pollution sources. So you can not only get a good snapshot of what's going on right now within the watershed, but you can also kind of go back and see good examples of trends and changes over time within the watershed and the pollution source impacts that might be occurring. So once we kind of know what's going on within the watershed, the second main part of our growing areas program is our bacteriological water quality sampling program. This is where we actually go out and measure water quality directly through water quality sampling. So right now we've got a little over 900 sampling stations that we actively monitor throughout the coast. We are required to sample each of these stations at least 6 times per year in order to kind of assess what the baseline level of pollution is at each of these locations. So that equates to about 5,500 water quality samples per year. Obviously, there are a lot of different potential contaminants that could be impacting water quality in our shellfish growing areas, but it would be super expensive and time consuming to test for all of them. So instead, we've chosen one particular indicator species to monitor for, and that's our fecal coliform bacteria. That's the primary thing that we monitor for within our program. So fecal coliform bacteria are only naturally found in the digestive systems of humans and other warm-blooded animals. So if we find it in growing area waters, that's an indication that either human or animal waste is present in the environment as well, and that other harmful bacteria, more harmful bacteria or viruses or parasites that might be associated with human or animal waste could be present as well. The other advantage of fecal coliform bacteria over some other potential indicator species is that we only need to collect small water samples, about 100 milliliters, in order to run the test, and it can be cultured relatively cheaply and easily within the lab, which is a big deal when you're running several thousand water samples per year. So we use a method within our laboratories called the multiple tube fermentation method with A1 media. Each of these test tubes contains basically a nutrient broth that has just the right mix of amino acids and salts and sugars to encourage the growth of fecal coliform bacteria. We'll take different dilutions of our water quality samples and put them in these test tubes with the growth media in sets of 5. So you can see the right-hand set of 5 test tubes all has 10 milliliters of sample in there. The next set to the left has 1 milliliter sample in each test tube. And then the third one that's behind these has a 0.1 milliliter sample within each test tube. We'll put the test tubes into a warm water bath for 24 hours. As the bacteria, if they're present in the sample, kind of grow and reproduce, they also let off a gas. You can see in some of those test tubes, or in all of them, we've got smaller inverted test tubes on the inside. If bacteria are present in that particular subset of the sample, it'll produce gas during that incubation period. That gas will collect in that inside test tube, and we'll use that as a indicator that it's a positive result. We'll count the number of positive test tubes in each set of 5 for the dilutions, and then we've got a chart that basically tells us what the total number of bacteria are in that sample. This particular lab test generates a type of a result in units of most probable number, MPN. Because we're not doing a direct count of bacterial colonies like we might do for a plate count or something like that. Instead, this is more of a statistical estimate based off of the dilutions and the results that we see of the amount of bacteria that are present within the sample. We've got a pretty strict water quality standard in order for areas to be open to shellfish harvest. So you can see at the top, using basically the last 5 years of data for each sampling station, so the last 30 samples. That location needs to have a median or a geomean of MPN of less than or equal to 14 fecal coliform bacteria, or an estimated 90th percentile of less than or equal to 43 in order to be open to shellfish harvest. The table on the bottom left there, you can see this is the highlighted column on the left-hand side of that table is the amount of fecal coliform bacteria contained in 1 gram of waste from those different types of animals or humans. The highlighted column on the right of that table is the amount of grams of waste on average that those different types of animals release. So you can see that it does not take very much. Takes, one person not cleaning up after their dog in their backyard and having it rain and run off. You can very quickly and easily exceed that bacteriological standard. So the areas that are open to shellfish harvest and meeting these standards are very pristine, in my view. So we take the shoreline survey information, we take the bacteriological sampling data and some other data that we generate along the way, and we use that information to set our shellfish harvest classifications. You can see the different classifications represented on this map here of Newport River and Bogue Sound. The dark blue areas are our approved areas. These areas are generally not impacted by any significant sources of pollution and fall well under the threshold for those different bacteriological standards that I was mentioning earlier. The dark red areas are prohibited areas. These areas might not meet those water quality standards or might be subject to a lot of known pollution impacts. So we have them off limits to shellfish harvest. The kind of hash marked areas you can kind of see down there, maybe near the causeway and near the port. This is our restricted areas. These areas don't generally meet our standards for safe shellfish harvest for direct consumption, but they're not so terribly polluted where we've found that through the shellfish relay process, the shellfish can be moved from these locations, held in clean waters for a period of time to kind of flush out the contaminants, and then they can be made safe for human consumption. And then the last two classification types, which I'll cover in the last part of my talk here, are the light blue and pink areas. These are our conditionally managed areas. So what we've found through our sampling and through other research that we've done is that, as you all probably already know, stormwater runoff can have a significant impact on water quality in a lot of our shellfish growing areas. And in particular, significant rainstorms can cause a spike in bacteria levels within the different growing areas that could render the shellfish temporarily unsafe to eat. This is just one example of sampling results that we have from Newport River at 2 sampling stations in Newport River, kind of showing at the beginning where those sampling stations are all the orange line on that chart is that 14 mpn threshold for kind of safe samples. You can see at the start there, before any rain events, those samples are meeting or below that standard. But then as soon as we get a rainstorm that dropped about 1 to 3 inches of rain within the watershed, you can see those bacteria levels start to climb and then start to recover after the tides have had a chance to flush out those contaminants a little bit. But then we get another rainstorm a couple days later, and it bumps those bacteria numbers right back up. And kind of that trend continues through subsequent rainstorms until finally about 5 weeks later, we're finally back down below the threshold for safe harvest again. So, because we recognize that these rain events with associated stormwater runoff can cause public health concerns with the shellfish on a temporary basis, we've set up management plans in each of these growing areas where we've seen these impacts, where after a certain threshold of rain is reached in a 24-hour period, we'll automatically close the growing area to harvest, and we'll leave it closed until we have the opportunity to go out and collect samples there again, showing that it's back down to below those safe harvest standards. We've got 22 growing areas throughout the state that have conditional management plans, totaling about almost 43,000 acres of shellfish growing waters that are conditionally managed. And our rainfall thresholds currently range between the extreme low of 1 inch in a 24-hour period that causes closure up to 2.5 inches. When you start to get above 3 inches, then we have broader, more emergency style closures to some areas. These conditional management closures, while they do help to keep some areas that are marginal open to harvest as frequently as possible, they do still have a large impact on shellfish harvesters. This chart is a little complicated, so I'll try and explain it here. But obviously on the x-axis is the years, and on the y-axis is temporary closure days. The blue bars are our annual rainfall across each of those years across the North Carolina coast. And then each of the individual lines represents one of our more popular and frequently used shellfish growing areas. So the dark orange is Lockwood's Valley River, the gray is Stump Sound, the light orange is Newport River, and the blue is North River. And this shows the number of days each of those years where at least a portion of that growing area was temporarily closed to harvest. So you can see that obviously it's very dependent on the annual rainfall, and obviously the more rain, the more temporary closures you have, since rainfall is the trigger. But you can see that even in the drier years like 2019, you still have over 100 closure days in Newport River and Lockwood's Valley River. In 2018, which hopefully we never have to repeat again, you saw almost 250 closure days in Newport River. So, you're losing a large percentage of the year where you can harvest from those areas because of the impacts of rainfall and stormwater runoff on the water quality in these areas. And it also, does add a significant amount to the workload that we have to do in order to sample to get these areas back open. I mentioned just for routine monitoring, we have to collect around 5,500 samples per year. On average, we have to do another 14 to 1,500 samples a year just to reopen after these conditional closures. So it can have a big impact on shellfish industry and on us as a regulatory agency. And with that, I will kind of wrap up. That's a quick summary of our program. There's obviously a lot more detail to it, but I'm happy to answer any questions now, or my contact information up on the slide if you want to reach out later to learn more about what we do.
You got a business card?
I do.
May I have one?
Yeah, I'll grab it from my office.
Okay. I got a question. How many on-site wastewater systems do you think, or have y'all studied, or failing in the state of North Carolina?
Well,
what kind of percentage of them? How many of them already? How many is failing?
I don't have the numbers off the top of my head, but on a typical survey, we'll maybe find 3 to 5 within a watershed that might be failing. In some of the more rural areas, we might see a higher number, but on the typical basis, they tend to function pretty well, at least when it comes to not surfacing and running off. There may be underground issues that we don't kind of have the opportunity to check on, but as far as surfacing, it's not a huge problem, but it is a consistent issue in that we'll always find a few.
Okay, well, how about the wastewater treatment facilities? Now, how big a problem are wastewater treatment facilities?
That has improved a lot since I started working here. I've worked with shellfish sanitation for almost 20 years. When I first started here, there was a lot of issues with wastewater collection systems in particular, but also the quality of the wastewater treatment plants along the coast. There's been a big effort in recent years to improve both the collection systems and the wastewater treatment plants themselves. All of the treatment plants in Carteret County for sure, and I think elsewhere that have discharges to coastal waters have all been upgraded within the last 10 to 15 years. And same with the collection systems for the most part. So we see a lot less issue now than we did maybe 15 years ago with treatment failures and overflows from lift stations.
Well, most wastewater treatment facilities are in the very bottom. I mean, because they all operate off gravity.
That's right.
And so they're sitting beside the creek.
They are.
So when you have a rain event, there's a lot of raw sewage sometimes gets spilled. And I've always heard that they actually have the ability. I do not know this, and I would like to know if they have the ability to dump a percentage of raw sewage. If there's some number that they can, we can dump 5 gallons today, boys, are they allowed to dump raw sewage?
Plants in North Carolina are not. There are some parts of the country that have older wastewater treatment plant systems where it's a combined system where all the stormwater from the city as well as the wastewater are combined into one and go to wastewater treatment plant. So when you have big rainstorms in those systems, you have a huge slug of water going to that wastewater treatment plant and they just can't handle it. And so it's better to let it bypass for a little bit so you can get control over the vast majority of it. The systems here are not set up that way. They're solely wastewater dedicated. There were historically a lot of issues with infiltration into the system from stormwater, but a lot of the efforts to repair those systems have been focused primarily on reducing that infiltration of stormwater. So you don't have the same kind of bypass issues that you do see in other parts of the country here. The treatment plants here also, since they've been upgraded recently, they tend to add a lot of extra capacity when they upgrade the plants so that they can count for future growth and not have to go and redo the plant again almost immediately. So there tends to be a lot more extra capacity in these newer plants for handling kind of extra slugs of flow versus what you used to see.
But that's where the fecal, the fecal coliform, that's where that mostly comes from, correct?
Right now, probably not. I think the wastewater treatment plants generally produce a pretty clean effluent and they have very strict limits, particularly when they discharge to coastal environments that they have to meet on a daily basis. I think a lot of it probably a lot of it's probably animal based. And then, you've got specific areas where it might be pets, it might be septic systems, it might be issues with wastewater systems in particular regions, but it's not that I would say it's not on a regular basis the primary driver of the contaminants that we see.
Great question though.
It's all for me.
Also runoff.
Runoff in general, yeah.
Runoff is carrying.
Right. Yep.
Well, everything that you're talking about is coastal.
Yeah.
Okay. All the water, a lot of the water at the coast is coming from somewhere else,
Right.
non-coastal.
Right.
And I'm not so sure all these wastewater treatment facilities further up the creek are doing as good as the ones down here are.
Yeah, I can only speak
Okay.
with familiarity with the coastal treatment plants that we visit and assess. They're very well maybe, there's like Havelock, for example, has a lot more issues with some of their collection system with frequent overflows, but that's far enough upstream and in our prohibited areas where does not impact our shellfish growing areas. It impacts water quality for sure, but not in our shellfish growing areas.
Well, I happen to live on Falls Lake, which is one of Raleigh's water supplies, and it is fed by Ellerby Creek, and there's a wastewater treatment facility sitting on Ellerby Creek, Durham.
Yeah.
And some days you can cross Ellerby Creek and it's black. Some days you cross Ellerby Creek and it's not.
Yeah.
Okay.
Yeah.
So
yeah,
and Falls Lake recreational areas stay closed to swimmers nearly the whole summer.
Yeah.
So they got thousands and thousands of people in recreational areas, but they're not allowed to go into water because the beach areas are closed due to the amounts of fecal coliform found in the water.
Yeah. Yeah, all of my context is solely coastal.
Okay. Okay. Well, all of that's coming down to noose. So
Yeah.
that is the noose.
Yep.
So
And that was one of our issue papers. And one of the reasons was because DWR was concerned about it, mostly the collection rates. And it's such a large issue on a statewide, even on our coast still, because there's so many peaks that we don't even see. So subtle problem and a lot of money to the Division of Water Infrastructure to fix that collection infrastructure and the plant. The local community have to apply. There's several different plots, but it's really so hopefully it's going to get better. WR has a website, those interactive maps that shows like where the wastewater plants are. Bill data on there.
And I would have thought that a large rain event would dilute more than concentrate. I was just thinking opposite on that. I was,
The issue is, I mean, obviously you've got clean water coming from the sky, but then when it hits the ground, particularly in areas that are more developed and have more impervious surface, that water can only kind of trickle into the ground so much before it starts to run off. And when it runs off, it also carries with it any contaminants that might be on the ground. So like I was mentioning earlier, got somebody who hasn't cleaned up after their dog, that dog waste is going to be hit by the rain and kind of carried off into the nearby ditch or the nearby creek.
Right.
And, you know, when you have enough of those sources kind of coming in after a rain event, you can see those spikes in bacteria levels. If it were not for the stormwater runoff, then certainly you'd see water quality improvements because you'd just be adding good, clean water. But everything it runs over, it carries with it.
But we test just mainly for the fecal coliform,
right?
Because that's a much cheaper test.
Yes, it's a good indicator of the presence of other potentially more harmful things, but is a little bit easier and cheaper to test for.
What's the next test? What's the more expensive test?
Well, I don't know what these tests are. I mean, I think it's called a BOD or something.
I mean, there are a variety of different water quality metrics, but a lot of them are more related to, I guess, water quality in general versus human health, and water quality. So we're kind of, as an agency, we're focused on making sure that, you know, the shellfish are going to be safe to eat.
Right.
We're not as concerned with like nutrients and that sort of thing, because that's going to be more of a environmental issue within the watershed versus human health issue.
Like toxins would be carried in that runoff as well from the
Right.
roadways, like petroleum products. So
Yep.
they're not testing for that because, you know, you can drink toxic stuff and you won't die right away, you know, but you may slowly get cancer. It's just very indirect correlation, but so it's just, that's expensive to test for.
But would if you find high concentrations, would you want to do a more expensive test for other types of, because it's an indicator that these things are there? Would you at that point want to do a more expensive test for other types of
So we
contaminants?
typically rely upon our shoreline survey data, so that pollution source identification information that we collect on land to identify areas where we might have concerns about things like oils or gases or heavy metals. And we'll actually kind of put prohibited buffer zones around those locations. So because, you know, we don't have the capabilities to do a lot of that testing, instead we'll just kind of, we'll identify the risk through our shoreline survey and the closure buffers in place that way through testing results. So.
Do you live on the lake?
Huh?
Do you live on the lake?
Well, nobody lives on the lake. Corps of Engineers has a buffer. I can hit a golf ball to it.
I'm glad you brought it to the table. I just.
You know,
I got one.
Yeah.
So if it's stormwater runoff, you know, for our coastal areas, we need to get it from the inland. With our coastal areas in the runoff, what would you feel is like number one, number two, number three biggest polluters of our water? In regard to that, is it dog waste, pet waste? Is it septic system failing? You know, which would be the source? Is it human, dog?
Seems to be a lot more animal. You know, there are concentrated areas where it's certainly kind of more human influenced. But on a widespread basis, it seems that it's probably more animal.
When I was harvesting oysters in December, we had this weird little blue, like bird flu come through, and there were tons of dead birds all through where I was harvesting. And it really bothered me, and I wasn't gonna harvest from there because there's this dead carcass sitting on top of the oyster beds, and there's so much more than we recognize that's out there that's
just nastying up our waters. And I don't fish with my dogs. I don't do any of that. I'm just me on my boat. And I just, I can definitely see that bird sickness that came through just really opened my eyes to how quickly someone could get sick. And so I stopped harvesting during that time. Sucked for me financially, but didn't make people get sick.
Yes. Longer term implication.
Yeah. There's a lot out there that we have to take into consideration.
Follow up to what you said about animals. Does that mean hog farms? Is that how that translates?
Oh, yeah. I'm sorry.
We don't typically, I don't think, see issues from hog farms down here on the coast. That's more of an inland issue.
Rivers?
Yeah,
Okay.
the river habitats they're far enough upstream where we don't see impacts in the shellfish growing areas that we manage.
I felt that during Hurricane Florence. I couldn't work for 6 months.
That was a lot of water coming down,
Yes,
especially after that with Hurricane Michael.
it was bad.
That water.
It was really bad.
Yeah. It was high tide.
Dead fish all over I-40, dead hogs.
Yep.
Everything.
Yep.
Lisa?
Yeah, sure. I feel like we could probably answer that a little bit better a year from now because we just got some funding to be able to do bacteria and DNA source tracking for pretty much all of the New River, which has a heavy concentration of concentrated animal feeding operations, specifically hog and poultry, at the headwaters of that river. And so what we want to understand is if those, that bacteria coming from those hog and poultry farms are getting down to the estuaries. So we should have a pretty good understanding or a better understanding than we do now of that a year from now. And we're also looking into, I mean, this kind of goes a little bit off kilter from the human health aspect of it, at least at this point, because we just don't know enough to understand the impacts to human health of the levels that we've been seeing in surface waters of things like PFAS and heavy metals in this particular water body. But we're also seeking funding to do PFAS and heavy metal testing of both wild caught and farm raised shellfish in that area, and we're hoping to be able to get that on track for next year. But we have known to see bacteria that could be coming from hog farms in the estuary and downriver. And like you said, with big significant storms, you're definitely going to have that. And considering that we did have a harmful algal bloom in the lower part of that river last year, it's definitely of a high concern of ours to be able to get a better understanding of that.
Those results will come out to the public. Would that be a correct assumption?
Yes, absolutely.
1 year from now. And may I ask another question, if that's okay? So I appreciated and enjoyed your analysis of your rains in a larger public health impact. I was wondering just the opposite. When we have a bunch of days or even weeks, sans rain, and now the saline, amount of saline in the water goes drastically higher. And I know you've been referencing the Newport River. There are some places that are quite tidal, probably don't have that issue with high saline levels. And there are other sections of the Newport River that are probably not a recipient of a good tidal change. So when there is more saline in the water, can you please share with us what happens then to the coastal environment and the oysters, et cetera, from a public health standpoint?
We certainly see when we have dry periods like we did for the last several months before the recent rains, you certainly see pretty dramatic improvements in at least the bacteriological water quality of the coastal environment. The salinities definitely go up and bacterial levels certainly go down. One thing I didn't mention when I was just in the interest of time earlier, but those pink areas there, those are our conditionally approved closed areas. So these areas typically don't meet water quality standards, but we have found that in circumstances like what you're mentioning, when we don't have rain for a long period of time, that these areas can temporarily clean up enough to be safe for harvest. So after some additional both water quality and shellfish meat testing in those locations, we can open those up temporarily to harvest while the kind of drought conditions, for lack of a better term, persist.
So would you say always when there's more saline in the water, it is always more beneficial to human consumption? Is that a direct correlation?
Generally, the higher the salinities, the more ocean influence and the less kind of stormwater runoff and freshwater influence you're seeing. So generally, I would say there is a correlation, but it's not always, it's not something that rises to the level where we could use it as a management indicator.
Okay.
But in general, the things that create good water quality also lead to higher salinities in coastal land.
Thank you.
Yep.
Just got one thing real quick. So as the resident meteorologist, I worked with Andy for a while on different things. If you guys want to feel like you can contribute to this, there's a good citizen science program called CocoRoss. And I just brought one flyer. If you guys want to check it out, I'll leave it on the table. But it stands for Community Collaborative Rain, Snow, and Hail Network. This data is used daily by Andy and the crew here to look at points of rainfall. So if it could be close to meeting a threshold, sometimes they look at the raw data in CocoRoss, but they also use obviously our rainfall data from NOAA. But if you live, especially in an area that's not populated, or if you know of folks that live in areas that are sparsely populated, we'd love to get more volunteers in these areas because it can be quite valuable to get that rainfall information. Not only is it helpful for them, it's really cool for us at the Weather Service because it's weather history essentially. The next Michael or Matthew or whatever, Dorian, we've had so many recently, they're all going together in my head. But the next big storm, you're forever part of that weather history. So it's kind of cool from a weather perspective. But anyway, I just wanted to share that with you guys.
Yep, we use that site on a daily basis. And in fact, if people are interested in signing up to be CocoRoss volunteers, we have a set of rain gauges we can hand out for free to people who are interested in volunteering.
Could I ask a follow-up about that? Is that linked also into the personal weather station network that is mediated through Weather Underground and all the other such networks? Is that tied?
No, it's completely different. So CocoRoss data, it's a manual observation with a plastic grain gauge. You check it once a day in the morning, or you can make big reports if you want to. If it's a big enough event, you can make multiple reports in a day. But what you're talking about, the personal weather stations are all automated, and they're still used. It's part of the overall aggregate collection of rainfall data that Andy and Shannon and the folks here look at. But it's not the same as a human observed rain gauge. When it rains really, really, really hard, like what we saw in Florida a couple of days ago, the automated gauges will not capture all that because it'll spill over. It can't. Wait, essentially what it is, it's a bucket and it tips. Yeah. And so eventually there's so much rain it can't catch up, can't keep up with heavy rainfall events. So you can't beat the human observed manual rain gauge.
Yeah, I just wondered about that question because as personal weather stations proliferate, so I've got 4 or 5 in my neighborhood here in Raleigh that are really interesting to look at, and particularly given how variable rain precipitation patterns actually are. And so if you're using just the RDU number or something else, it really doesn't reflect that well on the overall density within the watershed. I just wondered how those weather and how those are being used in characterizing rainfall patterns.
Yeah, it is. I don't want to speak for Andy here, but what it is they pull in radar data, they pull in the point source data like a personal weather station or CocoRoss, and there's essentially an aggregate output put out, and then they can overlay it with their shellfish harvests to determine whether an area might be close.
All right, we're on to water quality standards. Did you say how? But you mentioned that for your.
Yep.
Make sure I get it. And then it's get, I mean, I'm out there every day, I get reported too.
There we go.
First I need to, right.
You voiced it well. You held on.
Just hold on.
You can have it if you want. Or, for whoever wants it.
Doug, how's your back feeling?
I've got a heated pad back here behind me, so I'm better.
Good.
So, got it. All right. All right, well, thanks, everybody. I've been up since 4:15 this morning. I drove up to Chapel Hill, talked about Falls Lake. Another meeting, gave a few talk, come back down here, so I'm kind of punchy. So stop me if you have any questions. Don't let me just keep going. Yeah, if there's any questions at all, just stop me and ask when you have the question rather than wait towards the end. Yeah, so I'm talking about water clarity and particularly a couple projects that I've been working on in collaboration with Albemarle Family Code National Estuary Partnership, as well as Water Resources. So as part of the CHIP amendment, the seagrass was a pretty big focus of the 2021 CHIP amendment. This picture here on the right is it's showing this extent map. All the green in there are places that seagrass are either currently or have in the past existed. This becomes important later, so just keep this idea that we know where seagrasses have been. To be honest, there's not been a great accounting in the past. The current efforts for really monitoring where seagrasses are and how that's changed over time, we're getting better at it. But we're losing seagrass. First of all, there's two different kind of types of seagrass we have in North Carolina. We have a high salinity seagrass that you can see if you went out to the Outer Banks or out here in Bogue Sound. It's generally short grasses. They end up spending a lot of their energy, putting energy into their roots to hold them in these energetic environments that we have down here. They need a lot of light. They need about 20% of incident sunlight to grow well. Then we have low salinity seagrasses, things that you would see like up the Neuse River, or those types of seagrasses tend to be canopy forming, so they'll grow up off the sediment bed, and they can be 4 or 5 feet tall at times. They can grow in fairly deep water, extend their canopy way up. They don't tend to put a lot of energy in their roots. Most of their energy is up in their leaf structure. And because they grow up high in the water column, they don't need as much light. So the CHIP amendment, in the CHIP amendment, there's a specific emphasis on making sure that there's enough light available for these two different types of seagrass, the high salinity that require a lot of light and the low salinity that require less light. And within the CHIP, there was a literature review done. A lot of this was based on work that was done in Chesapeake. And this target water clarity levels were written down as, this is what we should shoot for. So for the high salinity seagrass, the CHIP amendment proposed that the seagrasses need 20% of light down to a 1.7 meter depth. And that's based on work in the Chesapeake and also the depth level that we see seagrasses growing down to in North Carolina's high salinity environments. And then for the low salinity, 13% of incident sunlight down to 1.5 meters depth. So light, when it comes into the water, it declines exponentially with depth. And you can describe that, how fast it declines exponentially with a rate constant. And that rate constant, just by convention, is used the letter K. That's just why. I have no idea why, who started using K, but it is. So when you see this K, which is going to come up, that's all it is. It's how fast that exponential decay happens. And so if you work out the those targets, 22% to 1.7, that works out to an exponential decay of negative 1.36 for the high salinity. Sorry, negative 0.89 for the high salinity and negative 1.36 for the low salinity. So the bigger this number is, at least the absolute value of it, the faster the light is being attenuated, or the poorer the water clarity is. So I'm dealing with optical models of water clarity. So for me, I take that 22% to 1.7 meters, and to me, just 0.89 is a lot easier. Just that's the clarity of water you need. And so this is what is currently being proposed as a standard for water clarity for North Carolina's estuarine waters. There's a process called the Nutrient Criteria Development Plan. And so this is through Division of Water Resources. And this targets that were proposed in the CHIP are now going through the process of being considered as true water quality standards for North Carolina. And right now, the thinking is that this would apply to all the estuarine areas with historic SAV. So that map I showed you before, these new rules would apply to those areas. And these clarity standards would then apply to a median value during the growing season, which is May to October. That's what's currently being considered. So my role or one of my roles in this process has been looking at, okay, so right now we know what causes the light attenuation. It's really three things. It's the amount of algae that are in the water. We typically measure that as chlorophyll A, which is the green pigment that's in all plants. We use that as a proxy for how much phytoplankton is there. It also happens to be what absorbs light for plants. Also, that's the process. It's absorbing light for the seagrasses. So we use chlorophyll A as a really effective measure of how much light the phytoplankton or the algae are absorbing in the water. The other thing is just turbidity, how much sediments and detritus are up in the water. That can be really important. And the third thing, and I know you've all seen this, you drive across some of our coastal rivers and they're brown. It's tea color. It's the tannins. The colored dissolved organic matter that's in the water can be really important for determine how much light gets to the bottom in some of our estuarine waters. So this model, it's an optical model, and it's what I'm using to kind of investigate what's causing clarity problems, and then also where we need to set targets for things like chlorophyll A and sediments in the water to make sure we're going to have enough light for the seagrasses. This model takes those three parameters, turbidity, color dissolved organic matter, and chlorophyll A. You give them those three, and it can tell you what that exponential decay constant would be. Or you can turn it around, which is what I did. You could, if you know the target light attenuation value, then you can say, okay, where do I need to have chlorophyll A and turbidity? Those are the two things we kind of have some control over. We can control nutrients, that controls how much phytoplankton is in there. We control like runoff and erosion, that controls how much sediments are in the water. The color dissolved organic matter, it comes out of swamps, it's natural. I don't think we have much control over it. So we can control how much, or we can use the model then to say, okay, how much chlorophyll and how much turbidity can we have given a natural background color dissolved organic matter concentration and still have the light we want for seagrasses. So that's what I've done. And so if this model I'm showing here in this graph format, this red line is a line of constant light attenuation. This would be, for example, the 0.89 value for high salinity seagrasses. And you can get to that line. So anything kind of towards the origin would be good enough light. Anything up and out is not enough. So if you're at this black line, which means you've got way too much chlorophyll and turbidity, you could come back down to that line in lots of different ways. So you can set strategies for bringing down chlorophyll or bringing down turbidity to get you back down to light conditions that you want. So one of the things I was tasked with was looking at, okay, what's attenuating light in North Carolina waters? And currently
do our standard water quality standards for chlorophyll A and turbidity, are they sufficient to protect water clarity for seagrasses in the waters? So I was using this model that was developed up in the Chesapeake, and then postdoc back in mid 2000s used the model that was developed in the Chesapeake in North River. So he calibrated this model, was developed in Chesapeake, calibrated it for the North River estuary. So, and the idea being that it could then be used throughout all North Carolina's estuaries. And so before I just went and used the model, I wanted to make sure that was actually the case. Like, does it work for all North Carolina's estuaries? And then, so after I validated the model, which I'll get to that in a minute, but then I wanted to predict what levels of chlorophyll and turbidity we would need to meet those targets. And then if it didn't jive with our current standards, you know, I can make some recommendations about how the standards might be adjusted so that it would. And then secondly, I'm going to show some data that just shows like what the current water clarity is in North Carolina. I put together all the water clarity data we have, so we get an idea of what the actual current light climate is for seagrasses in North Carolina and where we're at in relation to those targets. So this is the validation data from high salinity waters for the places that we actually have measures of light attenuation. The observed K value, the attenuation is on the y-axis, and on the x-axis is the predicted. This is North River, so the model fits pretty well there because that's where it was calibrated to. As expected, the slope's near 1, and you know the intercept's really small. For Bogue Sound and Pamlico Sound, it's close. You know, we have a slope near 1, fairly small intercept, so it seems like it works okay for these other high salinity areas. In the low salinity areas, it didn't perform so well. It really underestimated the that light extinction coefficient. So this model that was calibrated in high salinity waters works well in high salinity waters, and it doesn't work well in low salinity waters. Something optically that's different about low salinity waters. And I'm working on a separate project right now to recalibrate this model for our low salinity waters. That's a project that's funded by APDEP, and I'm working with Division of Water Resources on as well. Yeah, so it worked great in high salinity, not so good in low salinity. So we can say something about what those thresholds of chlorophyll and turbidity need for the high salinity right now. We can't say anything about the low salinity. So the next thing I did was I tried to put together all the data sets that I have for CDOM, turbidity, and chlorophyll for high salinity waters in North Carolina. And this is it. This is all those data sets. And none of these data sets, other than the North River, had CDOM data. For these high salinity areas, the CDOM is not as important as the low salinity waters, but it is a little bit important. And I had to just estimate the CDOM based on the relationship between salinity. CDOM comes from freshwater pretty much, or freshwater inputs. So using a relationship I derived from in North River, salinity versus CDOM, I use that a model to figure out how much CDOM should be in the high salinity waters for that amount of salinity. Only thing I had. So I had all three predictors of light attenuation. And so what these figures are showing, the yellow dots are median values, all these cloud of data points. And to look at these thresholds, I made the assumption if I'm looking at chlorophyll A, that I can't change turbidity. So turbidity, I'm just going to set it at the median value. It is what it is. And then what would chlorophyll A have to be to bring me, how high could it be if I'm, you know, good currently, or how low would it have to come back down to if I'm, you know, out there somewhere where there's not enough light? Where could chlorophyll A have to be to come back to that line of constant attenuation? So these numbers in red and green are the turbidity and chlorophyll values that the model will predict will get you what you want, where you want to be if you held the other value constant at the median, the current median. Yes.
For me, it looks like it's black. Maybe
The yellow dot is the median value.
any colors except black.
It's just really hard to see.
It might be hard to see.
I could see it on the screen, but you can't see it up
Look,
there.
if you look at the bottom left panel, you'll see, let me go to this one.
Make it larger.
There, there,
Yeah.
there's one. See, those are the,
Okay.
there's-
Like a larger yellow dot. Yeah, I didn't see him at first either.
yeah. So if you just kind of like broadly look at these numbers, a turbidity threshold is somewhere around, a chlorophyll threshold is somewhere like 10 to 15 micrograms per liter seems like a reasonable number. And a turbidity value is somewhere around 5. Nephelometric turbidity unit seems like a reasonable value. And we compare that to the current standards, chlorophyll A is 40 and turbidity is 25. These aren't exactly apples to apples comparisons. But I can tell you that so these current standards are really a do not exceed value. So I'm comparing a median value from these thresholds to actually a 90th quantile value, which makes it just really confusing. But I've worked this out statistically. I can tell you that
Believe you have.
even try to like make an apples to apples comparison, these standards are still not protective. They need to come down significantly. And so they need to come down to these median value of these thresholds.
Impressive information.
So that's kind of where we're at. Current standards aren't going to cut it for the high salinity SAV. I need to recalibrate this model for the low salinity and then do the same thing. I think that the story is going to be similar. I'll be really surprised if the current standards are going to get us where we need to be for the low salinity SAV habitats. The next thing is, where are we currently at? So we have a lot of water clarity data in North Carolina from an instrument called a Secchi disc. Secchi disc is basically a pie plate. You hang it down in the water. When it disappears, you record the depth it disappeared at, and that is a Secchi depth. So it's a visual, it's using your eyes, and it's a measure of water clarity. However, it doesn't necessarily measure the light attenuation at the wavelengths of light that plants need. So it's an indicator, but it's not a great measure. After the CHIP amendment, we started realizing like, wow, there's really not a lot of this data that we're saying we're going to use as a clarity standard. We better start measuring it. So in collaboration with AppNet and Division of Water Resources, we put together a special study, and Division of Water Resources for the past year and a half measured this light attenuation function. And so that's what I'm showing here. These are values from these 10 stations in Alcohol Sound. And this black line here, see that? The black line here is that proposed standard of 1.36 per meter. The median values are the red bars. The median values in all these figures are the red lines in these boxes. And then the boxes are the interquartile range. The whiskers go out to some, I can't remember. But pro standard is the median value. So you can just really look at the red lines, and you can see that for the most part, these stations don't, they're not going to meet this criteria. Yeah, so there's some thinking about, okay, like, yeah, what are we going to do? In some cases, like for the Pasquotank River, for example, which isn't on here. Oh, shoot. Yeah, they didn't sample that. I did. I'm not showing my data here. Okay. Pasquotank River would be off this chart. It is like awful. I mean, there's tea color in North Carolina's estuarine waters, but Pasquotank River is calm. It's like 6 per meter. So and it never, I mean, this is natural. It drains out of the Great Dismal Swamp. It's a natural condition. So there's some thinking about what we do about waters like this.
That's tannic acid.
Yes, just the tannins. Yeah. And it's, so yeah, we're gonna have to think about where this, but saying that, remember what I said initially, that this is gonna apply to where seagrasses have historically lived. They never historically lived there anyway. There was never any light at the bottom there. It was all,
It's
all,
natural that way, right?
yeah, exactly. So, yeah, these are things that are being thought about through this nutrient criteria development plan process though. So if we look at rivers like Pamlico River and Neuse River, the upper parts of these rivers are not currently meeting this clarity standard, and the lower parts are. And there's SAV in the upper parts of some of these, mostly at shallower depths than the target depth. But I know, like, I've duck hunt a lot in the upper Neuse, and there's places where I can find SAV way up past Newbern, and it's usually in water that's typically about that deep. And same with the Trent River. Anyway, that's kind of the trend as far as these rivers. Upstream's not going to meet this clarity standard. Downstream is. Those records really have been, just like the last year and a half. There are some places where we have longer records, and I'm showing these areas here because we can kind of look at over time how attenuation changes. So this is the upper Neuse River from the Neuse River Modeling and Monitoring Program that Hans runs out of IMS. Been doing it since '94, and you can see here a clear kind of wave to these light attenuation data as river flow changes. So similar to fecal coliforms, the freshwater inputs are bad for water clarity. It's a combination of sediments,
Runoff.
nutrient inputs for phytoplankton, as well as more of that color dissolved organic matter. It's all three probably. That, more runoff leads to all three things. And these upper stations, you can see, almost always they're these are well above that black line. The upper stations just aren't going to meet this clarity standard. Pamlico Sound, this is for the high salinity clarity standard. So you can see that value is now at 0.89, and the values are like all over the place around that line. So depending on how the clarity standard is written, as far as like how many years out of X can it go over without triggering a violation, this could be in compliance or it could be in violations, and all that stuff is still being talked about, be worked out. These are some areas that are really high salinity near Middlemarsh and Shackford Banks. And these, we didn't actually directly measure these values, but it was calculated using that optical model. And see, near the inlets, Shackford and Middlemarsh, most of the time they're clear. Yeah, near Beaufort Inlet, the water's clear, and most of the time it's good enough for the SAV. And there's a lot of SAV there too. Middlemarsh has got some really nice seagrass beds. And then Pamlico Sound out at Ocracoke, again, it's right there at the. And then Bogue Sound off the IMS pier, right here. It's generally not meeting that clarity standard. There's no SAV right there by the dock to speak of. Really energetic, and it's kind of deep. Lots of boat wake. I don't know if how representative this is for Bogue Sound as a whole. It's probably, my guess, it's probably not. But that's the data we have. And so this is measurements that were made using the Secchi disk, mostly. Some of these data are actually measured light attenuation values. Some of them are modeled using the biooptical model. These little symbols that, whether they're circles or squares, tells you what the data you're looking at actually are. And then the color tells you what percentage of the target light is actually getting to the bottom. So if it's twice, that's really good. That 200% of that, so 44% would be getting to the bottom. That is twice the target in these high salinity zones. And really the only place you see that is Core Sound. So
Is where?
Core Sound. Core Sound is the gem of water clarity in North Carolina. It's the only place where it's like SAV look like they're getting everything they need light-wise. Everywhere else,
Because it's away from people and you have a lot of inlets.
I think that's exactly. Yeah, there's no major river. There's no major river directly going there, and you've got a few inlets. Yeah, and there's not a lot of.
Is that where the Middlemarsh is?
Yeah. Yep.
Okay.
Yeah.
So is that where the Middlemarsh is?
Middlemarsh is the southern part of Poor Sound.
Okay.
Well,
Well, not, Poor Sound technically ends at Barden's Inlet, which is Cape Lookout.
Mhm.
Everything
Above it.
above that, yeah.
Does the red say very poor?
The red is very poor.
Okay.
So if you squint your eyes, all you're gonna see is orange and reds. So that's not good.
Or no? All
Um,
right.
with a few greens sprinkled in here and there, so you can't really see this here.
Yeah.
Throughout everything, there's a few greens, and you could barely even see these little numbers that are beside each symbol. Those are like the number of observations.
Oh,
All those greens are like one. When they're sprinkled in amongst a bunch of red, those are usually like one observation that was done by the EPA as part of their coastal condition assessment. So it's a really low sample number on what must have been just an unusually good water clarity day.
we have been doing less sampling up there. I was in the sanitation lab today and they were talking about G1, G2, which is up in that northern region, and they just don't have the people to do it.
Yeah, so most of
sucks.
the data was collected by NCDMF
Okay.
as part of their trawl survey data. It's a mix of like every agency and program that I could put together, everybody, but the DMF trawl survey data was a big part of it.
Yeah.
And 915, we gave you everything.
Yeah, I'm sorry. Yep.
It's hard to get people up there,
120
Yeah, so.
especially with collecting data within 6 hours.
data. It might have had shellfish there.
It might have.
No, because you guys don't collect that second. They don't collect second.
I ask you too, so what about satellite data? So with chlorophyll A, you can get some.
You can see, yeah, it's hard in our waters because of the CDOM and the turbidity. It's really hard to back out chlorophyll. And it's also hard to measure, because of the chlorophyll, it's hard to measure CDOM very well. It's just they all gets really muddled. CDOM in particular makes it really hard to back out things like turbidity and chlorophyll because it absorbs so much light that not much light gets back to the satellite. So you're looking at, it's a signal to noise issue. And there's very little signal that comes out of our darker waters. So it makes it really hard to see, to get any kind of accurate measurements. It's something that a lot of people are working on, but it's really hard. And the darker the water, the harder it is. Yeah, so that's what I wanted to show you all. There's this ongoing stuff, I'm doing that, recalibrating the model. Been collecting the CDOM data and the PAR attenuation data that we're kind of lacking for the water. So we're putting together a better data set of that. Currently, most of estuarine waters aren't meeting these clarity targets.
Thank you so much for your research. This is eye-opening and fascinating. I'm excited to look at it more and get back to you. I'm sure we all have more questions to get back on you.
This is that, it's beautiful now, when it hadn't been before.
Yeah.
And that DWR has committed to doing the standard, but it's just taking them longer because of like what a bad picture it's painting, how to handle that.
Yeah, it's complicated, and there's a lot of really good people through the scientific advisory committee that are really thinking about how to best implement this. So this whole nutrient criteria development plan, there's a scientific advisory committee that's like looking at the science behind this and what's the best way to write the criteria so it protects the seagrasses.
Mhm.
The other side, there's a criteria implementation committee that looks at the practical aspects of, okay, so we have this new standard. How are we going to monitor to make sure we're meeting the standard? How are we going to propose to actually get there from where we're at currently?
Yeah.
Kind of thing. So
We appreciate all of their hard work. It's impressive. It's hopeful. Seems to feel hopeful.
Doug's got a question.
Yeah, duh.
Hey, I'm just wondering, this sounds like an opportunity to develop sort of a new overlay on the citizen science paradigm. And I'm just wondering if this depth isn't adequate, whether you guys have or are developing a replicable citizen science application that could be very widely applied. Here, obviously, AppNet and H in its first iterations had fairly large citizen science water quality volunteer overlays mediated through where East Carolina, I think, did it. I just wondered, in particular up in all of the lower salinity areas, there's lots of people who live around, who fish around, who do whatever. So if you had a QA/QC technology better than, more applicable than Sechi disk depth, it seems like you could be fairly easy to spread that. Is that part of your thinking?
Well, the issue is the equipment to actually measure light is pretty expensive. So it's kind of beyond what you could ask a citizen science, a few $1,000s for a
Yeah,
light
I'm
meter.
sure that's true now, but the question I'm asking you is whether there's an innovation opportunity to consider outside the box new gear that would be citizen science appropriate and accurate. In other words, you're not looking for the same kind of professional replication that you guys get, but whether you could imagine something that would do the trick. I mean, for that matter, if you look at both at drone and sensor technology going through the roof, underwater drone technology developing rapidly, whether there might be something that one could have people pay for themselves that could create a new data stream that might. And I'm thinking about implementation in the future, not supplementing your data set now.
I think it's possible. I mean, I could imagine that you could make a really cheap light sensor with today's technologies. I just haven't seen one.
Is there anybody that comes to mind in your back pocket, Doug?
Well, I'll tell you what I was actually thinking about is what is an agreement that we had cut with fishermen, and particularly with the Professional Association of Fishermen's Groups in Peru, where they, well, first of all, we've got wave gliders, so autonomous and semi-autonomous vehicles going out to do certain things at or near the surface. And actually, we have some underwater working on corals in Belize, but they were talking about using ship fishing vessels as vessels of opportunity, not too different than Ferrymon and things like that, but with newly developed cheap sensors deployed on nets and feeding back data in near real time. And if you sort of think about a whole fleet, then you've got a big set of new cheap sensors. Because I guess what I was really thinking about is how fast the price of sensors has come down. And
I do know of some cheap sensors.
yeah,
Now I
and
think
so
about it.
wouldn't it be fun to try something out like that and get? I bet that would pay for that kind of, maybe even EPA would pay for that kind of new test to see if you could take your new sensors and get people to be trained well enough to give data that's good enough, maybe not perfect, professionally perfect, but good enough for a network of information that you might not otherwise be able to afford. Anyway, just something to think about.
No, it's a good thought.
Yeah, that's fun. I'm excited about that. That'd be neat. Any public comment this evening? I don't think we have anyone. All right, anybody else have any issues they want to talk about?
This is very off topic from what we've been speaking about tonight, but timely in that this happened last week. So another dolphin had its tail cut off, consequently died, and washed up, and Marine Fisheries with one of the doctors in the area did a necropsy. Although it's not a big leap to obviously figure out how it died and possibly even who, human-wise, made it pass away. And I was wondering if there is any way to with the help of software that is available insofar as figuring out wind activity, current activity, tidal activity, and with the necropsy, knowing approximately when this animal died, to figure out what possible commercial fishermen may be in the area, to question him or her or they as to was anything seen or or just to investigate that. So again, very off topic for tonight, but it just happened this past week, and I think the necropsy was done, took 3 hours. I think it was on Saturday that it was done. So I don't know if anybody has any more knowledge or thoughts or what can be done to not have this incident happen yet again. I know that we have created, we meaning the greater we, nets when loggerhead turtles get stuck in them, in commercial nets that they can swim out. I don't know if, again, the greater we are working on any cetaceans that may end up in these nets if there's an escape route, or if that would just lose too many of the fish. But I throw that out only because of the timeliness, and Marine Fisheries did the assisted in the necropsy.
Anna, if I may.
Yes.
I think, obviously if anyone else here has any knowledge they would like to add in, I certainly think that's something that I can make a note of, and I can try to follow up, with our folks that were more involved in the necropsy process, and see what other information we have. We can get something back to you.
I couldn't ask for more. I was telling Paula earlier today that about a couple of weeks ago, I came across a dead dolphin, and of which I took many photos of and reported the position. And they weren't able to find it, although that dead dolphin, as clear as possible could be, was a shark attack, and that you could see the 2 bites taken out of this animal only about yay. Quite sad, but that would appear to be 100% natural, as opposed to just a slice of the tail would very much appear to be unnatural. Let's just put it that way.
I was out there this morning and, the people that I was on the boat with alerted me that there was a dolphin that had been caught in a net and was struggling, and we couldn't find it. And when we were working together to gather the data that we were gathering, we heard a puff because we all know what the ocean sounds like. And we heard that dolphin puff, and we looked out there for probably like 30, 45 minutes. We drove our boat around, we were looking for him. DMS is very, very aware and cautious of that situation, and it is just as troublesome to the people that are working for DMS daily. It's a tearjerker out there. It's intense. And
2 of my passengers were in fact crying.
yeah. It's we want to fix those problems.
So that would bring us to a law enforcement issue.
I'm not sure. I don't know I don't know how it happened. I just was collecting data on the boat and heard that was an issue with the dolphin, and we were looking for it, that's all. And it broke all of our hearts. We were all very upset about it because we were like, how is it growing into that net? It's traumatizing. So our heartaches. Very hard.
Well, thank you for that understanding. And again, I know it was very off topic.
It's okay.
Downer for tonight, but thank you for letting me bring that up.
Yeah.
Just in other fisheries north of here where there's more whale population, Some of the nets that they use up there have pingers on them, which are like a deterrent, for those mammals, like, so it's just something that's out there for technology. It's just, I haven't seen it in the use in the national fisheries south, like the Cape. Like I've seen them up north though.
Yeah, and I wonder what net it was caught up in, and it is devastating.
Yeah, whether it's a gill net.
Yeah,
Sure,
we don't
whatever.
know, but it still sucks, and we're definitely all looking out for it. I literally thought I heard it today. We got all excited. We're like, can we save it right now? And no, and that just made us even better. So the heartache is there.
Maybe it was the one that was in my creek this morning.
Oh, God.
Better. See,
What's her name? Daphne? Let's save her. Daphne. God. Oh, it's heartbreaking. I gotcha. Thanks for bringing it up.
I have one more. I bring it up, this is like the third time I probably brought it up, is just mitigating strategies for new construction and repair. We were talking about water clarity, talk about water quality, and it's definitely something that, I'm working on a project for living shorelines. So it's very disheartening that we can't, if I'm in a closed area where my property is, I'm legally not allowed to put oysters out there for water quality and habitat purposes, legally allowed to do it now.
Is it because of ratios to?
It's because of the concern that someone may eat the oysters, which would be located within the closed area. So my concern is I want to do it out of a habitat water quality concern, not necessarily out of I want to go eat some oysters out of my backyard. Yeah. Maybe someday, if there were enough oysters, it'd make it an open area, but I don't foresee that happening.
So are you hoping for more leases in your area? Is that what you're saying?
No, I just I want to change to go through the chip to say, hey, if you are in a closed area and you want to put oysters in, into a closed area, whether it's in bags like you would do for a traditional lease, it's not allowed currently.
Well,
Well, it's closed for a reason.
Yeah, I don't want to eat them. I want to
Okay.
just
Just
have
improve water quality.
water quality as a citizen.
Yeah,
Okay.
but I think the point is that is habitat for lots of
area
other,
over a period of time.
yeah, but it's habitat for lots of other species. And so if you can improve the quality of habitat for other species using oysters as living remediation, why not? As long as they're prescribed from harvest and contamination of the foodways.
Right, so I can't go by spat.
That is frustrating.
But you were talking about like a UDOC situation, weren't you? Brought it up
No,
before?
and I can use my own property. I've got my pier, and so they came and they built it. They did construction damage. They trampled some marsh grass, like obviously I put a 60-foot extension into the public trust and there's no mitigation. So if I was to do a seawall, for example, or a living shoreline, there is nothing legal that I can do other than living shoreline, which would, in theory, attract oysters to set on those places.
Gotcha.
But I can't legally jumpstart it.
Gotcha.
I can't go buy seed oyster
You
currently.
couldn't transfer wild oyster?
Correct.
Oh,
Not legally. And that's what I'm hoping that the chip would, modification, the more I say it, maybe.
Well,
That would-
we've been talking about the relays and I'm not,
Yeah, it's going backwards from a relay,
it would be going backwards, but
taking it from a good area to a closed area.
if it's for the benefit of the water, it's something to look at. It's definitely on the table.
That's on our list.
Yeah.
I keep bringing it up, maybe.
It's on the really list.
You can put some.
Yeah. Closed areas aren't bad places for oysters. They're just bad places to source oysters from for human consumption.
Exactly.
They add to habitat value of those sites anyway.
Yep. Yep. All right, so. Any other? We can adjourn the meeting.
But I've had a really great time talking with you guys. Should we go on? No, we shouldn't. We will be concise. I really appreciate all of you so much, so much. And I'm, again, sorry to not recognize folks that were not here in our presence. It's nice to see you and be with all of you. Thank you for your input and your information, and let's get an email train going so we can all stay in touch. And I'm really excited about what you are up to and any way that I can help.
On the AppNet website, there's a report that I put out, I guess it was a year ago now, that covered a lot of this in more depth.
It's fascinating.
It's on the AppNet website. Yeah, you can look it up, and can't remember exactly what the title is, but it's about water clarity and SAP.
Nice.
It's not
Thank
too
you
hard to
all
find.
for all your hard work.
In the following email, can we get some of those links?
Yeah.
Just so we could find
Oh,
it,
yeah.
you know, just like I was looking for the chip earlier today. Making sure
I want one too.
I think that would be good for all of us to be able to reference.
Appreciate all of your knowledge and passion, you guys.
Thank you.
Thanks.
You have a motion to adjourn or anything?
Oh, yeah, there's a motion to adjourn. Do I have a second?
Approved.
It's funny you said that.